The overall goal of this methodology is to study the import of nucleus-encoded proteins into chloroplasts with a particular focus on the effects of stress.
This method can help answer key questions in the chloroplast field, such as how chloroplast protein import is regulated. A particular advantage of this technique is that it allows for the effects of stress on chloroplast protein import to be assessed. Though this method has been developed for use with the model plant Arabidopsis thaliana, it could be modified and adapted for use with other systems such as crop plants. Demonstrating the procedure will be Qihua Ling, a postdoc from my laboratory.
To begin, first grow the Arabidopsis plants as described in the accompanying text protocol. Then, when the plants are 8 days old, perform a stress treatment. Gently scrape the plants from the agar plate by hand and place them into a sterilized flask of liquid MS medium containing the stressor. Placing additional plants into a similar flask without stressor as a control might also be necessary depending on the nature of the experiment. Cover the mouth of the flasks with sterilized foil and allow the plants to grow for an additional 2 days on an orbital shaker with gentle shaking. Prepare a continuous density gradient by first mixing 13 milliliters of percol, 13 milliliters of 2x chloroplast isolation buffer, and 5 milligrams of glutathione in a 50 milliliter centrifuge tube. Then, centrifuge the mixture. After centrifugation,